Hello, this is Alice Formiga of eOrganic, and I'd like to welcome you to today's webinar on organic growing media, how can it affect the health of my transplants, by a team of researchers from Purdue University. eOrganic publishes many free articles, videos, and webinars about organic farming and research, and you can find all of them on our website at eorganic.org and on the eOrganic YouTube channel. The recording of this webinar will also be posted on the eOrganic YouTube channel within 1 to 2 weeks. This webinar will last about 45 minutes, and then we'll have 30 minutes for questions. If you have a question at any time, feel free to type it into the Q&A box on your Zoom control panel. To find the Q&A box, just hover over the bottom of your Zoom screen, and there should be a black bar with some controls. And if you click on the Q&A one, that should open it up. You can also use the chat box, and that will let you choose to send a message either to just the panelists or to all the attendees in case you have something that you'd like to share with everyone in the audience. So now I'd like to welcome our 4 presenters from the Department of Horticulture and Landscape Architecture at Purdue University. First, we will hear from Wenjing Guan, who is an assistant professor who's a horticultural specialist doing research in sustainable specialty crop production. Then we'll hear from Liz Maynuard, who's an associate professor and serves as an extension specialist in vegetable production, conducting applied research and developing information resources to improve vegetable farming. Petrus Langenhoven is an extension specialist, and he specializes in hydroponics and vegetable crop production and designs research and extension programming around new technologies, high tunnel and greenhouse management, and open field vegetable production. Finally, Lori Hoagland is an associate professor and soil microbial ecologist. The long-term goal of her research and teaching program is to support the specialty crop industry by identifying practical approaches to managing microbial communities that can help plants acquire nutrients, suppress diseases, and prevent uptake of heavy metals. So with that, I'm going to hand over the remote control to our first presenter, Wenjing Guan.
Like Alice said, my name is Wenjing Guan. I'll go first, give an introduction about organic growing media. Liz Maynard will present our research results about seedling performance in different media. Followed by Petrus Langenhoven, he will talk about using organic fertilizers with the medium. After that, Laura Hoagland will discuss medium microbial interactions. Lastly, Liz will summarize what we have learned through the project and give practical recommendations. Thanks. Respond to the fast-growing organic industry in the U.S. Certain certified organic operations that report to produce organic transplants are also steadily increasing. Using data from USDA Organic Integrity Database, we noticed certified organic operations that produce transplants increased from about 1,200 in 2015 to close to 1,800 in 2018. Majority of the operations reported they produce transplants for on-farm use only, or for on-farm use and for sale. Only a few operations specialized in producing organic transplant only for sale, and likely they are for specific crops such as tobacco or hemp, not for general vegetable crops. Next, please. Thanks. Majority of organic vegetable producers are growing their own transplants. This situation determines that there's a large variation in organic transplant production in order to meet individual producers' specific needs. For example, some organic producers prefer to use peat pot or soil blocks rather than using standard plastic trays. Some producers may grow tomatoes to a relatively large size before transplanting, especially in the early spring. And some producers grow transplants in an indoor condition for better controlling temperature in early spring. This condition often have a no or very limited natural light. Next slide, please. No matter what system organic producers use to grow their own transplants, growing medium play very important roles in ensuring success of this system, as it provides plants aeration, water, and nutrient. Unique for organic growing mediums that the medium must meet USDA National Organic Program standards. Ingredients have to be natural materials or synthetic ingredients that are on the national list defined by the NOP. Let's take a closer look at the common materials in organic growing medium. Some of these materials are the same for both conventional and organic medium, such as perlite and vermiculite that help with aeration for the medium. Peat and coconut coal or other byproducts from cocoa industry are used to help with holding water. The materials that commonly included in conventional medium but cannot be used in organic medium are synthetic fertilizers and synthetic wetting agent. There's a large variation in nutrient sources in organic growing medium. Next slide, please. Okay. We did a OMRI search using keywords transplant container medium and potting soil. This search led to a total of 364 products from 197 manufacturers. Those products include the ones used for growing transplants in small cells, also included products for raised bed, houseplants, larger pots, and in hydroponic production. We want to know what are the materials commonly included in organic medium that provide nutrients. We then separated the products that clearly identified their nutrient sources and created this finger on the right. Stand out from this finger is compost. There are a total of 71 products contain compost. Not every product specifies the compost source, but the ones we could find the information use chicken manure compost, cow manure compost, lobster compost, compost from grain waste, or plant-based compost. In addition to compost, other materials providing nutrient in organic medium include animal or plant-based meals. Next slide, please. Such as alfalfa meal, kip meal, fish meal, bone meal, soy meal. These meals are great nutrient sources. Soybean meal is a good source of nitrogen. Fish meal is rich in both nitrogen and phosphorus. Kip meal provides trace elements. We also found products include worm castings and bat guana. They are both great nitrogen sources and release nutrients faster than most compost, providing readily available nutrient source for plants. Dolomitic or calcitic limestones are usually included in the medium to adjust pH. Gypsum, potassium sulfate, green sand are also included in some medium to provide nutrient. In this search, we also noticed mycorrhizal, humic substances, and biochar are included in some of the mediums. Next slide, please. We know nutrient management aspects for conventional grown transplants has been extensively researched and largely optimized. But there's little research on nutrient management in organic transplant production. What makes the nutrient management in organic media complicated is that, first, compost, the most widely used nutrient source, have varied nutrient content depending on raw materials. In addition to compost, there are multiple organic amendments available with different nutrient combinations. Secondly, seed germination. Seed germination is particularly sensitive to salinity. Too much compost or other organic amendments can lead to increased salinity and interfere with seed germination. Thirdly, seedling growth. Seedling growth is in a relatively short period. Synchronized nitrogen mineralization and other nutrient supply from medium with transplant needs are very important, but poorly quantified. Lastly, post-transplant plant growth. Transplant health may affect crop yield, especially early yield, and whether microbes and transplant stage affect post-transplant growth is largely unknown. Addition to this puzzle is physical, biological, and chemical interactions in the medium. Compost and organic amendments not only play important roles in providing nutrient to the medium, but also affect water holding ability and aeration of the medium, and support microbial activities that have a direct impact to nutrient availability and potentially other functions such as disease suppression. So our research surely cannot answer all these questions, but hopefully it would help you better understand organic medium and help your production. With that said, I will lead it to our next speaker, Liz.
Okay, Liz, I'm going to hand you the remote control and hopefully that'll work. So just click on the screen there and hopefully that should work. And make sure you're unmuted.
Thank you. And thanks, Wenjing. Um, as Wenjing indicated, there are so many different growing media available. Deciding which one to use can be difficult. We did this project to learn how much difference the choice of the media makes and how the plants grow, and how common tests of chemical and physical characteristics could help growers identify media likely to work well for them. Now, for growers who already have identified media that performs well, what we learned might not be that useful. But for growers who are just starting out or not quite satisfied with the media they are using, we hope that it'll help you select media. Our first trials compared media without any added fertilizer, and in a second set, we added fertilizer during seedling production. In addition to the chemical and physical characteristics of the media, we know that biological characteristics of the media and an influence on biology can make a difference, and so we took the opportunity to look at those effects also. In the experiments presented today, we looked at just 7 media. We had 3 crop types, tomatoes, cucurbits, and lettuce, and we did our trials at 3 Indiana locations. Okay, there we go. So these are the 7 media and the codes to abbreviate them that we'll use throughout the presentation. Uh, we had 5 media that contained compost: Johnny's 512, Morgan Composting, Premium Flour 201, Penn Valley Potting Soil, Seed Catapult from Ohio Earth Food, and Vermont Compost Fort Light. And then 2 without compost were the ProMix MP Organic and the Sunshine number 1. These 2 without compost were the least expensive. We were paying on the order of less than $15 for a 60-quart bag. The most expensive was on the order of $45 for a 60-quart bag. But I have to say, price can vary a lot depending on whether you're buying in bulk or the size of the bag, and also on how far you have to ship. We did not get any compost-based media from the West Coast because we're in Indiana. Shipping was expensive. Even shipping from places like Maine or Vermont are fairly expensive for us. So these are the media that we used, and you can see the ingredients. There were a variety of differences among them. We grew our, we grew our, sorry. We grew our seedlings in the greenhouse at 3 locations: northern Indiana, West Lafayette, and southern Indiana near Vincennes. We seeded directly into standard flats that were cut in half. Our original flat size was 72 cell for tomatoes, 50 cell for cucurbits, and 128 cell for lettuce. And we had different cucurbits at the different locations: cucumbers at Purdue in northern Indiana, summer squash in West Lafayette, and watermelon at the Southwest Purdue Ag Center. So we can think about performance of these plants in 3 stages: first, germination and emergence, then growth up to transplanting, and then performance in the field. So we saw differences in emergence right away. About a week after emergence, you can see nearly all have emerged in this media, none on the right. And by 2 weeks later, plants have come up in the media on the right, but from the variation in size, you can see that some are still coming up. So emergence occurred over a long period. If you plot emergence versus days after seeding, you can see these differences graphically. Total emergence is here. And then the speed of emergence, you can look at how long it took for 50% of the plants to emerge. So for this solid line graph, it took a little between 5 and 6 days for half the seeds to emerge. And for the dashed line, it took about 9 days for half the seed to emerge. So quite a delay in emergence. And then from the graph, you can also see how uniform the emergence was, how long it took for the seeds to emerge. On the solid line, that is pretty vertical, so that means they emerged over a certain short time period, fairly uniform emergence. The dashed line, it's more slanted, that means it's less uniform emergence. So ideally, we have high emergence, quick emergence, and uniform. In the next graph, I can show you results from all the media. There were certainly differences. This big graph in the middle shows the results for tomato. And one media definitely lagged behind the others. That line in red is the Vermont Compost Fort Light. It had less maximum emergence, slower emergence, and less emergence uniformity than the others. Several media performed well. Those were the Sunshine number 1, the Pro Mix Organic, and the Seed Catapult up here. We had similar results in the cucurbits also. So we had results of lab tests for a number of media characteristics, and one of the chemical measurements was EC, or electrical conductivity, also called salinity. It measures the dissolved salts. The salts could be essential nutrients for plants like potassium or nitrogen, or it could also be elements like sodium. And the media did differ in the EC measured before planting. And we found that EC had a strong relationship with emergence. So this is tomato emergence, the maximum emergence. The lower the EC, the higher the emergence. At higher EC media, we had lower emergence. And in fact, when we looked at the effect of EC on speed of emergence or uniformity of emergence, we also saw that lower EC was better. Now this slide has graphs for maximum emergence, speed of emergence, and duration or uniformity of emergence. And for all those things, for all the crops we looked at, lower EC was better. We had higher emergence, faster emergence, and more uniform emergence when the EC was lower. The general relationship between EC and emergence is something that has been known for a while. High salts are known to inhibit germination. And in media specifically for germination, it's sometimes recommended that the EC level be less than 1. We did also make lab measurements of media bulk density, pore space, water holding capacity, but those didn't help explain the differences in emergence that we saw. We did measure actual media moisture levels after seeding, and that did influence emergence in some of the trials. We had difficulty wetting some media at some of the trials. So here you can see a little bit of ponding on that media. Here also a little bit of water ponding, whereas with these other media, the water went right in. When we had difficulty wetting the media and had lower water content, we did see problems with emergence. It's clear that if media does not wet easily, it's best to wet it before filling the flats. Let's move on to look at seedling growth. There were obvious visual differences. The 7 media are across the top here, and then each row is a different trial. These are all the tomato trials. And it's pretty clear that 2 media, this PMPO and Sun 1, produced smaller plants than the others. You can see similar pictures of the cucurbits here. We have the cucumbers in the top 2 rows, then the summer squash, and the bottom row are the watermelons. And again, that PMPO and the Sun 1 had smaller plants. Also with the lettuce, the PMPO and the Sun 1 had smaller plants. In the lettuce trial, we did have an 8th media, the Vermont Compost Fort V. So we can look at a graph of dry weight. And here we have dry weight of tomato and cucurbit plants at about 3 to 4 weeks after, well, a little, in some cases a little bit longer, but at about the time when we would be ready to transplant. And it's pretty clear there are differences among the treatments. We looked for a relationship between the growth measurements and chemical characteristics of the media like EC or pH, but nothing really stood out. But it was clear that the media containing compost produced larger seedlings than the media without compost. One of the compost media didn't do quite as well as the others. Now, for many producers, the real test of performance will be how plants perform in the field. We planted the tomatoes out in non-certified but organically managed areas at the 3 locations in Indiana and followed them for at least 4 weeks to see how they grew and how they started to set fruit. In these photos from the field, you might be able to see size differences. These were 2.5 to 3.5 weeks after planting in the field. This is the PMPO plots, and this is the Sun
stuff.
One plots.
It's actually
The we took weights of these
Wenjing,
plants and can see that result in this graph.
medium stuff.
The seedlings grown in the compost containing media were larger 4 weeks after transplanting to the field than the seedlings not in compost containing media, although those in PMPO and Sun 1 had caught up a little bit. The larger plants generally flowered and set fruit earlier. This graph shows the bars for the 7 different media, and then the different colors of the bar represent the percent of plants that had fruit in red, or open flowers but no fruit in yellow, or buds but no flowers, and that would be or in green. So the Vermont Compost Fort Light was the earliest to flower and set fruit, followed by the other compost containing media, and then the Sun 1 and the PMPO were the last. We looked for relationships between that fruit set and the chemical measurements of the growing media that we took before seeding. And again, EC turned out to be important. The plants grown in media with higher EC flowered and set fruit earlier. This is the percent of plants that had fruit by that date, and plants grown in media with higher EC had a higher percentage. In a case of one particular nutrient, nitrate nitrogen, there was a similar relationship. The higher the nitrate nitrogen, the higher percentage of plants that set fruit by that 4 week after planting. But there were some outliers, so there's clearly more to understand with that measurement. So because the EC level is associated with the nutrient level in the growing media, and based on other observations we made on the plants, it suggests that the nutrient level in the growing media is behind the big differences in performance that we saw. A saturated media extract test is a standard method to get information on media EC as well as on the level of readily soluble nutrients. And it seems to be really useful for characterizing these media. So we think that would be one test that growers could do to assess media. The results that you see here include the EC that I showed you before. Here's the nitrate, and then pH and other soluble nutrients. And these were obtained by sending samples to a commercial lab for a saturated media extract greenhouse media test. This is the type of test that could be helpful in assessing different media. But just looking at the differences in nutrient levels that you see in these media and the results from this trial without fertilizer, it raises the natural question of how using fertilizers would change the performance of these media. Petrus will fill us in on what we learned about that. I will turn over to Petrus.
Okay, I'm going to just hand Petrus over the remote control. If you could unmute Petrus, that would be great. And let's see. And okay, if you click on the screen, you should have the control.
Thank you, Alice.
Welcome.
As Liz mentioned, in our first round of trials, we noticed that seedling growth was affected by media type. Media containing compost produced larger seedlings than media without. However, one of the composts containing media, M201, did not perform as well as the Johnny's 512 pen SCOE, and the VCFL media. In general, measurements recorded before transplanting revealed that M201, PMPO, and Sun1 produce significantly smaller plants with smaller stem diameter and lower dry weight. So we had to ask ourselves, how would transplants react if we added additional nutrients to the media? Will it improve seedling performance, or will it affect seedling growth negatively? We identified 3 methods for adding fertilizer: incorporate it into the growing media, apply it as a top dressing and leach it into the media through overhead irrigation, or apply it with your irrigation water. For experimental purposes, we selected a solid and liquid fertilizer. The question you might ask is why? We needed all 3 major nutrients to be present. There are cost implications for the growers. Growers might choose a fertilizer according to the existing equipment on the farm. Nutrients in the liquid fertilizer are easily available, while solids dissolve or release nutrients slowly over time. We selected Sustain, a slow-release nitrogen fertilizer, as one of the nutrient sources. It is a solid granulated material with a 524 NPK analysis. Sustain has a turcular and feather meal base and is OMRI approved. 90% of the nitrogen is slowly available. It also contains nutrients like iron, manganese, and zinc. Additional features include humic acid, organic matter content of 65%, a pH around 6.8, a CN ratio of 4:1, and a bulk density of 38. The second nutrient source was a concentrated liquid based on chicken manure and bone meal. The EnviroCure product has a 323 NPK guaranteed analysis. It is OMRI approved, but is listed with restrictions due to the presence of nitrate of soda, which is highly soluble. We mixed the 5-4 in at a rate meant to provide enough available nitrogen for seedling growth during the propagation period. We applied the 3-3 liquid fertilizer on the surface at 50 to 75% emergence, and then at 2, 3, 3.5, and 4 weeks after seeding. The fertilizer was diluted with water so that the final solution contained 500 ppm nitrogen. We fertilized aiming for the same amount of nitrogen, but ended up being a little short. Tomato seeds were germinated and seedlings were grown in 5 growing media: M201, PMPO, SCOE, Sun1, and VCFL. This picture was taken 35 days after seeding. Visually, you can notice that seedling growth was improved when additional nutrients were added. The substrates without compost, PMPO, and Sun1 produced the smaller seedlings when no additional nutrients were provided. PMPO and Sun1 reacted very positively to fertilizer application. Let's see if we can explain what happened during the propagation process. Here's a side-by-side visual comparison of treatments 35 days after seeding. The compost containing M201 media did not show response to the 5-4 fertilizer. Plants in both treatments look very similar, but slightly greener in the fertilizer treatment. The 5-4 application had a very positive effect on seedling growth in the non-compost containing PMPO media on the right. SCOE produced slightly more lush green plants with the addition of 5-4. Seedling growth in non-compost containing Sun1 media was positively affected by the 5-4 fertilizer application. Seedlings grown in the Vermont Compost Fort Light media on the left performed well without the addition of fertilizer. However, plant growth increased with the addition of 5-4 and 3-3. The 3-3 treatment appears to be a lot greener too. It seems the PMPO plant size produced even larger plants with the application of 323 without the lush green color of the 524 treatment. I would like to note that a different color system is used to identify treatments compared to what was presented earlier by Liz Maynuard. We only use 5 media instead of the 7 media used during the first experiments. We looked at adding fertilizer to media before planting or as plants emerged and saw effects on emergence. In the next couple of slides, we will concentrate on tomato seedling results. The graph on the right gives us an idea of how long it took to get to 50% emergence. Generally, fertilizer applied to media before seeding delayed emergence. The emergence delay was between 2 to 3 days. However, the emergence delay of the non-fertilized VCFL treatment was not significantly different from that of the fertilized treatment of M201, PMPO, and Sun1 media. The 323 fertilizer was not applied at this time, and therefore the results look very similar to the non-fertilized treatments. The graph on the right provides information about the emergence over 14 days, emergence uniformity, and total emergence. Let's look at the emergence uniformity. The steeper the slope of the curve, the more uniform emergence was. Non-fertilized media treatments showed very uniform emergence, except for VCFL. The PMPO 323 treatment received its first fertilizer treatment 5 days after seeding and showed similar uniform emergence as the non-fertilized media treatments. Emergence in the M201, SCOE, and VCFL fertilized media treatments was less uniform. This is the media that contains compost. Adding fertilizer did not affect total emergence significantly. Earlier, we saw results from a saturated media extract test that was performed on all media. The EC of the non-fertilized M201 and the VCFL media ranged between 3 and 4.5. In this graph, the box plot represents leachate data obtained from treatments using the pour-through sampling procedure 26 days after seeding and 3 days after the last fertilizer application. The box represents the variation in data, and the encircled dot, the means of the treatment. Tests of the media leachate showed that fertilizer treatments increased media EC, and this is probably why emergence was delayed. Emergence delay was more likely and larger in media with higher initial EC, for example, VCFL. The Sun1 media showed the least increase in EC. The magnitude of effects differed among trials, and that might be related to media temperature and or moisture content. It might be possible to reduce high EC by repeated leaching, but that isn't a good solution because one is just wasting nutrients into the waste stream by leaching. It's wasteful and potentially polluting. It would be better to choose a media with a lower EC when crops are germinated. We saw effects on the growth of tomato transplants in trays. The fertilizers made a difference, and for the low nutrient media, PMPO and Sun1 brought them up to performance similar to other nutrients. At 35 days after seeding, the non-fertilized PMPO and Sun1 media treatments produced a significantly smaller plant compared to all other fertilized and non-fertilized media treatments. In general, the 5-4 and 3-3 fertilizer reduced the decline in growth rate that occurred over time. We looked at seedling dry weight 35 days after seeding. The 5-4 fertilizer increased final plant size significantly for the non-compost low nutrient media PMPO and Sun1, and also for the compost containing media VCFL. The 3-3 liquid application seemed to do a little better. generally, it resulted in significantly larger seedlings in the PMPO and VCFL media, larger than the 5-4 fertilizer treatment. Effects were variable in other media, likely due to the delay in emergence caused by fertilizer incorporated into the media. The beneficial effects of fertilizer on plant growth in the greenhouse continued in the field. The figure on the right represents plant fresh weight data collected 4 weeks after transplanting. Fertilizer provided during transplant production generally increased plant size in the field, but effects were stronger for PMPO, SCOE, and SUN1, and not significant for M201 and VCFL. We also tracked crop productivity. Greater earliness, which includes bloom, production, fruit set, and yield, was observed with fertilizer treatments and followed increases in plant size. Fertilizer increased early yield for low nutrient media, PMPO, and SUN1. Non-fertilized M201 produced the highest yield. The table presents early and total cumulative yield data. The fertilized PMPO early cumulative yield was almost 9 times higher than the non-fertilized treatment. Similarly, the SUN1 fertilized treatment early yield increased 2.5 times. Fertilizer tends to increase total yield for PMPO and SUN1 and decrease total yield for M201 and VCFL, but variability was too high to conclude significant effects. I'm now handing it over to Laurie Hoagland.
Great, thank you, Petrus. Alice,
Okay, yeah, Laurie, you should have the remote now if you just click on the screen there. Yep.
Okay, yep, looks like it works. Okay, great. Okay, so now we're gonna switch gears a little bit and start talking about the plant microbiomes and whether they have a role or how much they could have a role in transplant production. So it has recently become clear, especially within the last 10 years in the development of new molecular tools, that plants are literally covered inside and out with microbes. So they're on the surface of the leaves, they're on plant roots, and they're also even living within inside of the plants. And so it might seem hard to believe, but like us, plants are actually more microbes than plants because the number of cells outnumbers those of the plant. And so there's a lot of interest now in trying to figure out what exactly are all these microbes doing and how are they affecting the plant. And so we know they can do a lot of things, right? And so first, we know what we know the most about is that some microbes can be bad. So these are plant pathogens that cause disease. There can also be some that are really bad, like foodborne pathogens, Salmonella, E. coli, that can attach to plants and negatively influence our health. But there's also many other microbes that are interacting with plants that can affect their metabolism, how they access nutrients, as well as help them to fight some of these bad and ugly pathogens that we don't want on a plant. Consequently, understanding and learning more about the composition of these plant microbiomes has a lot of potential in helping us to determine or promote plant health and productivity. And so in the future, we might see something like this, where just like a soil report, we can send off to a commercial lab and know what nutrients we're missing. Eventually, it's possible we could get to this soil microbiome report where we know which microbes we're missing and how to add them. We're not quite there yet. We do know there have been some good microbes that have been isolated and are applied as inoculants. But sometimes when we go out into the field environments, their survival can be variable, which we're trying to figure out why and how to better promote those. There's also many other good microbes out there that we're still unclear about what practices really promote those or how to promote those in plants. So one place to start in learning more about these plant microbiome interactions is to think about where these microbes are coming from. So we know that some microbes can be vertically transmitted from a mother plant to a daughter plant through seed. Others can be horizontally transmitted to plants from water, air, insects, even humans when we touch the plants. But soils tend to be one of the biggest factors shaping the plant microbiome. So it makes sense that potting media would also have a role in forming the plant microbiome, but there really hasn't been very much research at all in exploring this. So what could be in organic potting media? Well, we know that, as we've talked about in this webinar, there are some microbes that have been applied to these potting media as inoculants. And so one of those you heard about earlier are mycorrhizal fungi, which describes a symbiosis between plant roots and certain fungi in the Basidiomycota family. And so these microbes, they've been well studied and they're well known for their ability to scavenge nutrients in soil, solubilize them, and help plants take them up, most notably phosphorus and zinc. They can also provide other benefits such as increased tolerance to water and even pathogens. There are other microbes that have been isolated and can be applied in potting medias, specifically for their pathogen suppressive activity. So one of those is, for example, Trichoderma harzianum, which has a role in nutrient scavenging, but it's also widely used for its role in bioactivity or biocontrol against pathogens. And that can occur through a number of mechanisms, some being more direct mechanisms where the beneficial microbe is outcompeting the pathogen for space or nutrients or producing antibiotic compounds. But it can also occur through this indirect mechanism by helping the plants to scavenge nutrients, or like you see here on the lower right, actually inducing a plant's immune response. So strengthening the plant's immune response so that when a pathogen does attack it, it's better able to fight that pathogen off. And this mechanism is particularly important for foliar pathogens. So there's also likely to be many other microbes in organic potting media. And so we know a lot of the feedstocks that we've been talking about here, like sphagnum peat moss or compost, could be bringing their own microbes into the organic potting media. It's also likely that these media, the media that you're using, the fertilizers, are going to indirectly affect these communities just by how they're affecting plants, their growth, and in turn, how the plant is starting to release carbon compounds out of its roots to support microbial communities. So while we know this is likely, the influence, really what the community is or how this influences transplants is not well understood. And so that's why we wanted to take the opportunity as part of this project to look at what's happening, what microbiomes are living in some of these transplants, and how might they affect a plant's ability to withstand pathogen stress. And so I recruited some folks from my lab who did most of this work. So the pathogen that we wanted to use for these assays is Botrytis Cinnari, which causes the disease known as gray mold, which can be problematic in greenhouses and high tunnels at low temps. It produces many different symptoms, like this kind of rotting off of the plant stem or lesions on the fruit. We know there's not a lot of strong genetic resistance in tomato to this pathogen. And we know things like under and over application of nitrogen can promote susceptibility to the pathogen. And we also know from work in our lab and other places that there are beneficial microbes that can induce systemic resistance to this pathogen, like trichoderma, and reduce the disease. So we took tomato plants that were grown using these various potting media and fertilizer combinations that my colleagues described. And we inoculated them with suspensions of Botrytis Cinnari and then monitored lesion spread. Then we also collected DNA from the rhizosphere and root endosphere of these tomato plants and quantified the microbial communities using next generation sequencing and Chime bioinformatics software. So these are some of the results I'll walk through with you. This is the results of the Botrytis pathogen assay. And so the first point here, composition of the potting media did have some effect on susceptibility. And in this figure, the higher the bar, the worse. So that means there was more pathogen spread. And these red arrows are kind of pointing out the different potting media and how that affected pathogen spread. And so surprisingly, this Sun 1 did have lower disease than the other potting medias. We're not sure why. It's possible that media does have a silicon amendment that has been shown in some studies to induce a systemic resistance. So we also saw that adding fertilizer to the amendments, the potting media, reduced the spread of the pathogen. And that, if you look at these little green arrows, you can see that in most cases, there was a pretty dramatic drop in susceptibility of the plants to this botrytis. And then if you look at the circles here, these little boxes, you can see that we also saw differences between the two fertilizers. So the liquid one and the liquid, the E323, and the solid, the 524, where we saw differences, even lower disease spread with this different type of fertilizer. So next we want to look at the microbiome. We're still processing all of this data from these experiments, but I wanted to share some preliminary results with you. And this figure here shows two of the potting media. So this is PMPO and VCFL. And I just note one of them had mycorrhiza, one of them had compost. And this figure shows the microbiome, all the bacteria at the highest level, the phylum level. And so we did see clear differences in the community, even at the highest level, based on the composition of the potting media. So for example, in the VCFL, we saw fewer actinomycetes than the PMPO and more firmicutes. We also saw some changes in individual microbial taxa in response to the fertility amendment. So it's hard to see on this figure, but I wanted to just point out a couple of those in this next slide, that we saw increase in the presence of, in this case, the sustained fertilizer. And so we saw greater populations of Streptomyces, which is a type of, it's a gram-positive filamentous bacteria. I'm sorry, some of the words are kind of hidden behind these pictures. So this is what makes soil smell really nice, that geosmin that it produces. It's also very well noted for its ability to produce antibiotic compounds that can directly suppress pathogens, as well as has the capacity to induce systemic resistance. We also saw greater populations of Lysobacter, which is a gram-negative rod-shaped bacteria, also noted for the production of novel antibiotics and increasingly recognized for its biocontrol activity through various means. So what can we learn, or what did we learn from this study? Well, certainly as expected, just like in the field, tomato transplants are colonized by a very abundant and diverse assortment of microbes, regardless of whether they're actually being inoculated with specific taxa. We saw that the media composition and fertilizer does alter the composition of plant microbiomes. This is really exciting because when we think about trying to manage microbial communities and plant microbiomes for the health of plants, the transplant stage could be a very good place to start by seeding these transplants with good microbes before they even go into the field. So lastly, we did see that the type of potting media and fertilizer has a big effect on susceptibility to this pathogen that causes gray mold. We saw very strong correlations with the microbiome, but of course we need to do some additional studies to really say whether that was caused by these changes that we saw. But we can definitely conclude that proper nutrition is important. And so, having good transplants when you go out to the field is going to better prepare you and your plants to withstand pathogen stress. So with that, I'm going to turn it over to Liz to discuss the take-home messages.
Liz, I think you're still muted.
Hey, thank you. Um,
Okay, you've got the controls now.
Great, thanks. So, what have we learned about organic growing media and healthy transplants? Well, it's clear that the media can make a big difference in seedling growth and in crop performance. With some knowledge about the media, it's possible to adjust management practices to get good results. And the biggest differences in plant performances that we saw were related to the media EC or electrical conductivity. Too high and emergence was negatively affected, too low and the plants didn't grow well. With the low EC media, which is an indication of low nutrients, fertilizers could be used to improve the plant growth. And even in the high EC media, the plants could benefit, especially if those plants were grown for a longer period of time, they could benefit from additional fertilizer. The media that had higher EC or low EC media when it was fertilized led to larger plants in the greenhouse, in the field, and earlier production for tomatoes. And we didn't show it, but we did also see that we got earlier production in cucumbers and in lettuce. There was also indication that fertilizer reduced the disease susceptibility of the plants, possibly through impact on rhizosphere microbial populations and or the plant nutrient status that strengthens the plant immune system. And the growing media influenced the community of microbes associated with the plant roots, and exactly what implications that has, we still don't know. So I mentioned knowing the media is an important first step. And certainly one way to get to know the media is to look at information provided by the supplier. The information here is gathered from websites for some of the media that we used, but certainly more may be available by talking with a technical representative for the media. Oftentimes there's information on the type of the container, what crop or what crop growth stage media is suitable for. You'll notice that some here say they're suitable for seed starting, will not burn seedlings. There's often notes about the need for nutrients during production. And you'll notice that some of these say they have a small starter charge, the PMPO and the Sun 1. These are the two media that did not perform well for us without fertilizer. A starter charge means there's a little bit of nutrients there, but you'll need more to grow plants to full transplant size. Comments about moisture might say something about the water retention, whether it has a wetting agent or not, or whether it's important to wet that media before filling flats. So this information can be really useful, but realize that it's provided in part also for marketing purposes, and it's not necessarily standardized from one manufacturer to another. Comparing between manufacturers may be difficult just using this media. The saturated media extract test that we mentioned previously provides a standardized information, and we found it to correlate with some aspects of seedling performance, and certainly it's been found by others to correlate with that also. The standard procedures for this test are well documented. Commercial labs can perform it for a fee similar to field soil tests. Some media manufacturers provide example test results with product literature. In any case, testing media when it arrives at your operation using a test like this would provide useful information. If the nutrient content and electrical conductivity are low, you could plan to use an appropriate fertilizer. If that EC is high, it's probably not a good idea to mix fertilizer in the growing media before seeding. If you're germinating right in that mix, be sure not to let the media dry out, and maybe consider using a media that's lower in EC to germinate seedlings and then pot up into a higher EC media if that works in your system. If you're using fertilizer, use caution when mixing it into the growing media so you avoid too high a salt concentration. And waiting to apply until after seedlings have emerged can reduce the risk of delayed emergence due to those high salts. Of course, there are a lot of factors that go into what the best media is for a particular crop and a particular production system. And what we've gone over today is related differences in media performance in cell tray vegetable transplants, largely the differences in chemical properties of media. And then we found evidence for the media influencing the microbiome of the plant that could have long-lasting consequences in the field. But there's certainly potential for more understanding of media and for improving media and better matching it to specific situations if we can learn more about nutrient release, interactions among chemical, physical, and biological aspects. So to move forward, we'll need to have more research to tease out specific factors like that and combine that with the integrated knowledge that growers have from using different media and different practices in their production systems. Um, I'd like to thank everybody for joining today and mention that in addition to the 4 of us that you've heard from today, we had many other people working on this project and couldn't have done without the folks working in the lab or in the greenhouse and field and supporting the research facilities at Purdue. And nor could we have done it without the financial support from USDA and the Indiana State Department of Agriculture through a Specialty Crops Block Grant. Again, thanks for joining us, and that's the end of our prepared comments, and we should have time for questions. I'll turn it
Oh,
over to
yeah.
Alice.
Yeah, we do. And thank you so much. That was tons of information. And I'm sure that anyone who hasn't seen this message already in the chat box will be glad to know that we are planning to upload a handout of the slides to the Purdue website. And we'll be sending you to that link as soon as possible, hopefully today. And if not today, tomorrow. And that way you'll be able to look back at the slides because it was so much information presented. I'm sure people are going to want to go back and check what all those abbreviations mean. And we've also recorded this webinar, so we're going to be putting up the recording of the webinar on the eOrganic YouTube channel. You can just Google that, you'll find it. And it's also going to be obviously on our website as well at eorganic.org. So we have tons of questions, and if we don't get to all of them, we'll definitely be sending the presenters a log of all the questions so that we can follow up with you if needed. But we can run for another 30 minutes and answer questions. And I would think that we definitely have that many questions coming. I'm sure the presenters have seen them coming in thick and fast in the chat box here. So I'm going to just get started. Actually, in the Q&A box, if you could type them in the Q&A box, that I don't have to keep switching around from one place to the other, but I'll try to get to all of them and go back and forth here. So, okay, so one person wanted to know if there's a database somewhere where they can find out how many lumens their plants need to keep them from stretching. Does anyone know about that? Feel free to unmute all the presenters so that we can hear you. I think several of you are muted at the moment.
Hey, this is Wenjing. I don't know a database you could find that information. I think this will depend on several other factors, and there is for sure ways you could do to help the transplant stretch. In terms of the light, for example, like a blue light is to encourage more sturdy plants. So if you have a way to adjust the light spectrum using LED light, that's something you could do to help. And the strength of the night surely matters. I'm not sure of your production system. So that's my understanding. I'm not sure if I answered your question here.
Okay. All right. Okay, so this gets into the details of the study here. Were PMPO and SUN1 considered uncharged media, and could that explain the low EC, good emergence parameters, but low growth overall? Please unmute. Yeah, okay.
I'll say I am not exactly sure of the term uncharged, but they did not have a high nutrient content. The Based on the manufacturer information, they say they have a starter charge of nutrients, which usually means just a small amount. And and certainly that's what was shown in our tests, the SME test that we had done. And yes, I think that is related to the low EC and the poor performance without additional fertilizer and the rapid germination.
I had a question, and that was, is the EC something that can be found on the bag? Is that marked somehow, or is that something that you had to measure um, in your study?
I haven't usually seen it found on the bag. When I have seen it, sometimes a technical sheet might have information on it, like that says the range of typical EC,
Hmm.
but it might not have, like I looked at one for one of the media and it said from like 0.5 to 2. That's a fairly wide range. So, but I think once a media might be fairly, depending on the manufacturer, one media might be fairly consistent from year to year. So if you know what one media is, um,
Hmm.
if that's some manufacturers will plan to have a same EC in the same type of media over time.
I think
Okay. Hmm.
Alice, I would like to add, I think people also have to realize that EC is just a generalization of the nutrient content. It doesn't tell you how much nitrogen or potassium or whatever is in that media. So it would be best if the manufacturer doesn't tell you, to really find out, what is in that media before you use it.
Okay, yeah, there was someone, we had a number of questions about EC, as I think it may be a new term for some. One here, it says, can you elaborate on the description of EC? And did I understand correctly that lower EC resulted in faster emergence, but higher EC results in faster flowering and fruiting, such as the case with the Vermont compost FL mix? If my understanding is correct, then higher EC is more valuable despite its impact on a slower emergence. I think there's definitely a lot of questions about this. So.
Yeah, I can mention that a little bit more. So EC, it stands for electrical conductivity, and it's actually measured by measuring the electrical conductivity of the soil solution or of an extract of what's in the media. Because I don't know if if you go into salt water, it conducts electricity a lot faster because of all those salt ions. And so when they're measuring the EC of an extract out of a media, it's basically measuring how readily electricity is conducted through the media. And that is related to the dissolved ions that are in that solution. Some of those ions may be nutrients, the different nutrients, but some of them may be sodium. And some of the media that we looked at did have high levels of sodium, which isn't really considered an essential nutrient. So that's just what it is. Um, and yes, there is this, the high salt levels can injure germinating seeds and slow them down. High salt levels can also injure roots of seedlings that have already emerged and damage the roots and make them more prone to infection and so on. But it's especially can be damaging and depending a little bit on the crop on that germination and emergence. And so we did see with higher EC, we saw that delay in emergence and less uniformity. But that media with the higher EC also had higher nutrient levels, and there may have been other things going on too, but it did have higher nutrient levels, and that led to the higher growth and the earlier fruiting and yield in the field. So if you could get it germinated and the delay in germination was not too much, then the high EC seemed to be an advantage. One of the things to realize about EC is the level that the plant or seed experiences is related to the moisture content of that media. As that media dries out, the EC that plant or seed or whatever experiences is going to increase because there's less moisture around. So the salts are more and more concentrated in the little moisture that there is. So if you have a high EC media and you want to germinate in that, if you can keep that very moist, And we saw that in some studies, the germination may have been delayed a little bit, but the plants were able to germinate and get up and then grow well. I don't know if that answers that question, but yes, it is kind of a trade-off.
Okay, great. Did you measure the physical characteristics of the media, such as bulk density, total porosity? In my own remarkably similar work, I've found these to be significant factors. Some of these compost-containing mixes have very high bulk density, and the mixing and assembling technique also plays a big role in my experience, according to the commenter. Do you want to
Well,
make
I
any
can
comments on that?
say I know that those were measured, but and we did try to see if there was a correlation with the other measurements that we took, but there were not strong correlations. I don't think it's that they aren't important. It just may be that other factors had a stronger influence.
Hmm.
In our system, you mentioned the mixing and assembling technique. The mixes that we purchased, they were mixed and assembled by the manufacturer. So what came out of the bag, that was what they had. Of course, we did give some thought about how we put them in the flats. We debated about how hard we should pat them down and so on. So realizing that can influence the density in the flat. And was there in terms of mixing in the fertilizer, and when we mixed the fertilizer in before planting, um, uh, it's possible that in some cases we didn't get that fertilizer totally evenly mixed in, and that could also have influenced the evenness of emergence in those treatments. I don't know if someone else has comments about that. Lori, I know the measurements were done in your lab.
Yeah, so I think, as you mentioned, we did measure those things, and I think I agree it's very important. But I think all of the media that we were working with, you know, had levels that were in a good range. So perhaps that's why, you know, we didn't see as strong an effect on those physical properties as we did some of the chemical properties. But it's certainly a consideration if you're making your own media, to take that into account.
And maybe just one final comment on that. Heavier media, if you're dealing with shipping the media any distance, or if you are shipping the transplants any distance, the weight of the media, the bulk density of the media could be a factor in the expense.
Okay. What's the best method to measure EC? Can you use a meter directly into the mix, or do you have to measure what comes out, the leachate?
Okay, I guess I can say something here. At, Here I do use a handheld meter to measure EC. I just use a Feldspot direct soil EC meter. It's come out the actual numbers is not exactly as the numbers we have from the labs with an SME test. The number are not exactly the same, but the trend is the same. So when we use the handheld meter, we just follow the instructions of the meter, like we start and water the medium, like wait for half hour, and did the measurement. I think it can be it is useful, and it's definitely much easier compared to the connect the leachate, but that probably will require some experience to get an idea of what level you would see may have some problems. Like readily available some of the reference. Like we know EC can be measured using like 1:1, 1:2 water mix. So the recommendations you find from the website are not always works based on the method you use.
Hmm.
So in general, I think the handheld EC measure meter is very valuable, but probably need some experience to get a better understanding of the readings you get and with practical like meanings for you. Is that
Okay.
answer your question?
Okay, thank you.
Yeah.
Um, do you have information on the marketable yield versus the unmarketable yield, and were any differences found? I'm not sure how long into the plant's life the study was carried to.
Um, okay, I think the yield information presented here are I think 1 year is marketable, another year probably is the total yield. Um, one of the years our trial had a problem of tomato fruit worm
Oh,
and we actually get quite a bit of unmarketable fruit. So we are using total yield. It make more sense. But just my observation, I didn't see like the difference between the like any of the treatment affect specifically marketable yield or total yield.
let's see. We have a question about whether you can recommend any resources for making your own compost-based growing media.
I wanted to mention that we did not look at that, but there are some publications that have recipes for growing media, and the ones I am most familiar with are from ATRA.
From
There is,
ATRA. Yeah, okay.
yeah, there is one called potting mixes for certified organic production from I think from 2004, and I think there's also a more recent one.
Okay. Let's see. Was the Sustain 524 fertilizer always mixed into the media prior to seedling, or was it also used as a top dressing after emergence?
In the first set of trials, we actually used it, well, it was mixed into the media. And then once we observed the delay in emergence, we decided to do another round of experiments, which we didn't talk about in this presentation, where we actually applied it through several applications after germination. So, and we didn't get the same effect on the emergence delay in that sense. So, but we haven't discussed that result in this talk.
Okay. Do you know of any analogous work on growing media for woody perennials? I think several
I
of you are muted. So, yeah.
I'm not familiar with
Okay, good.
any.
Me too.
Anyone?
Nope, I
Um,
can't
Okay.
help.
Okay. Thank you.
I think if when I'm searching with all the organic mediums, some of the mediums do specified like for the perennial crop. Something I don't remember which medium is that, but I remember I saw it. Maybe maybe you could go through the. I don't know. I don't have a readily available recommendations here.
Okay. Do you know what is considered an, I think you maybe mentioned this earlier, but what is considered an ideal EC range for emergence and an ideal EC range for growth and yield?
uh, for so, and this information is taken from largely research that's been done in conventional production. So for germination stage uh, plugs, uh, some guidelines are that EC should be below 1. Typically for most crops growing on from somewhere 0.5 to 2, I think is typical for growing on. But then when you look at recommendations for specific bedding plant production, you might get into higher EC levels for specific plants once they've gotten up a bit, plants that are growing faster. So there is information out there for that has been developed in the context of conventional bedding plant and hanging basket production and so on. And then I know that in terms of greenhouse production, EC is often used to, and I'll let Petrus mention that, but it's often used as a proxy for nutrient content, right, in hydroponic production. Different
Yep,
situation.
that's right.
Okay. Okay. We have somebody who's curious about what's known about the mechanism by which EC impacts germination. So, why is that?
I
Why
think
does it inhibit germination if it's a high EC?
I think it's just related to water availability. Like high EC, it's difficult to have those waters infiltrated to the seeds
Hmm.
and that affect the germination. That's my understanding.
Okay. All right. Let's see. Yeah.
I do want to add for the EC. I see someone is, I think the question we answered before, but someone is asking, is that the high EC is more valuable considering the fast growth? I want to add a point. I think in most of the cases, it probably is with fast growth, flowering. But in the cases for transplant production, like if you have a specific timing for selling the plants or maybe the timing for transplanting, fast growth is not always a good thing. But better control of the growth rate of the plants might be more desirable. In this case, the lower nutrient medium, but with adding additional fertilizers might work better, so you have more flexible to controlling the growth rate.
Okay. Will you be publishing the full results of the experiment? This person is very interested in the best media for lettuce production.
We hope to publish the this work, and we do in terms of the lettuce production particular. We covered some of that in a presentation this summer. I think we have I think that webinar is online. I don't know if Petrus can you find that and put it in the chat box maybe?
Is that Liz?
The webinar for this summer where I
All
talked
right.
about the lettuce
Mm-hmm.
production. I think in there the lettuce I discussed the lettuce information there.
All right. I will get it quickly.
Thanks.
Okay, that would be great. Okay, meanwhile, I'm going to look down in the chat log here because there's so many questions coming in here. Okay, so when is the pivot point where lower EC is better for seedlings but higher better for transplants? I'm not sure what you mean by the pivot point, but maybe someone else understands that. Okay.
Mm,
Yeah, I don't
I,
know that there's really an exact pivot point.
Okay.
I think it's just kind of continuous. So the main but I think it's certainly possible. I mean, we've discussed it a little bit before. I think it's certainly possible to find a media that will, that you can germinate in and go on to grow transplants. And if you need more nutrients, you can add them later.
Okay. How might water source influence these results? This person has water with high mineral content in her region.
Liz, are you going to answer that?
I can start
Yeah.
if you like. I know that, I mean, we have high calcium and magnesium in hard water at some of the sites. And big influence of hard water is that well, obviously it can supply some of the calcium and magnesium that you need. So if the media does not have much of that, having it in your water makes a difference. The other thing is that hard water or high alkaline water will over time gradually tend to raise the pH of the media. If the media pH is already high to begin with, it's possible that the water you use will make it even too high, and you might see some uh, problems with the crop. Then usually the first thing we see with high pH is micronutrient deficiencies. The one media, we had one media that was quite low in pH, and if you're growing in a place that has alkaline water, um, you know, with that media you have more leeway until it would get too high.
Okay. Let's see, what is the shelf life of mycorrhizae introduced into growing media?
I guess I can try to take that comment. So yeah, that's a great question, and one of the challenges with a lot of microbial inoculants. And so I don't know off the top of my head really how long that should last, but when you do buy inoculants, there should be a shelf life on those products that's listed, and how you store those is really important as well. So many products containing mycorrhiza or other beneficial microbes need to be stored in cooler temperatures, and there is certainly evidence that after some time they will no longer be viable. So I have seen studies that where they've looked at commercial products and found that the mycorrhiza is no longer viable. So again, I don't have a specific time frame, but it is something to consider in not storing these medias for an extended amount of time.
Okay. Did root disease or germination respond to differences in media water holding capacity or porosity? Do you have any practical suggestions for evaluation of air-filled pore space for transplant media? That's two questions there, I guess.
The first one,
First,
maybe
yeah.
I guess I could jump in a little bit. So we didn't specifically look at any root-borne pathogens, although with the microbiome studies that we've done, I think we will see if those were present. I'll leave it to my colleagues to talk about if they saw evidence of that. But I think that's something really where the physical properties can have a big role, like bulk density. And so there are some kind of simple methods that you can use to look at bulk density and making sure your potting media has enough aeration so that it makes the plants a little bit less susceptible if those are present.
Okay. Which three medium in your study would you say were the top two? Okay, this says three. I think, I'm not sure if you're asking for three or two, but the top overall performers. Yeah, the top three.
I don't know why nobody wants to answer.
I felt this will be
Like we don't want to promote
another
any.
project.
Nobody wants to
company,
promote it. We're officially not promoting any products, but in the experiment.
Right. I think we decided from the first year's results, and Liz, you can comment here, but the Johnny's 512 mix was one of the better mixes, and that's why we probably decided not to use it in the second year, if I remember correctly. But as we can see from some of the results that we got, especially with the plant that we took to the field, and we got some yield data from it too, that Morgan composting premium flour did pretty well, actually, even without the fertilizer. It produced pretty good yields. Obviously, we need more replicated trials to verify that kind of data, but the VCFL seems to be quite a popular media as well. The problem with that media is that it's made on the East Coast, and it's a long way to ship it, and it is heavy. But a lot of growers use it, and we found pretty good results with that.
It is hard to say. I think the seed catapult from Ohio Earth Food also performed well in a lot of the trials. I just make another comment about the different media, because even those two media that weren't compost based, when nutrients were added to them, they did perform well. And for some situations, those non-compost based media may have desirable qualities. So it's really hard to say the best, you know. It's
right,
because it really depends on the particular situation.
Right, it depends on your needs for that situation.
Yeah, I agree with what Liz said. And another thing growers may consider is you have the time to fertilizer through the transplant production period, or you want to have a medium, just you only need to water it and then don't think of other things. So it all depends on the production system.